and my pleasure to share my studies on stem cell and their regenerative and therapeutic application. So my name is Ahmed Morsy Abdelbary. I work in the Department of Stem Cell and Regenerative Biotechnology at Konkuk University in Seoul. So here I would like to show the location of my university here in Seoul. So we have very beautiful lake here, and also there is a lot of college and school in our university. And here is located the Department of Animal Science that I work in. So I wish you invite you one time to hear my university. So first of all, I would like to give you some introduction about stem cell. So the story of stem cell started from sperm when it fertilized oocyte and then forming zygote, and this zygote divide again to 2 cell and 4 cell, divide again to 8 cells and morula. And the important thing, it form a blastocyst. So this blastocyst is very important because it contains ICM or inner cell mass. So from this ICM, we can obtain the embryonic stem cell. So important thing when you study stem cell, you have to know the terms like totipotent and pluripotent. So this inner cell mass, so zygote is a totipotent stem cell. For the inner cell mass, that the source of each embryonic stem cell is pluripotent stem cell. The meaning of pluripotent stem cell means these cells can be differentiated to all of our body organs like ectoderm and mesoderm and endoderm. So each lineage, each line can produce different organ and like muscle, like hair, like neuron. So that's very important. But using this embryonic stem cell is somehow not ethical. So if you try to use embryonic stem cells, you can have ethical issue. So instead of that, there is a very famous, discovery by very famous Japanese scientist. His name is Shinya Yamanaka, 2006. He successfully could uh, reprogram fibroblast cell from our skin by using very key reprogramming transcription factor like OCT4 and SOX2 and KLF4 and so on, using viral induction or non-viral induction. So there is some issue using this, is not our topic now. And then finally, it can produce induced pluripotent stem cell. So by producing this induced pluripotent stem cell, we can overcome the ethical issue of using the embryonic stem cells that derived from embryo. On the other hand, this part is called the pluripotent stem cell, including induced pluripotent stem cell and embryonic stem cell. On the other hand, there is also other important stem cell called adult stem cell. This adult stem cell can be obtained from different various organ and tissue. For example, can be obtained from bone marrow, and also from muscle tissue or adipose tissue, and also from fetal tissue. And then you can isolate. I can explain to you about that in the coming slides. But these cells is multipotent. It can be differentiated to limited number of lineage, as you can see. So very important to know the terms like totipotent, this kind like zygote. It can be formed all entire organism, and the pluripotent mean can be differentiated to uh, the three lineage endoderm, ectoderm, and mesoderm. It's like embryonic stem cell, and multipotent is like adult stem cell, for example, hematopoietic stem cell, and also oligopotent stem cell, like neural stem cell, and unipotent. It can also differentiate it to only one or single lineage, for example, spermatogonial stem cell. So here, this figure can show you the different classes of stem cell, including pluripotent, and also the terminology of that stem cell. Actually, for example, mesenchymal stem cell is very important example of adult stem cell. So actually, this mesenchymal stem cell is somehow confusing with fibroblast. So the terminology of this mesenchymal stem cell is actually getting changed from 1986. It was identified by German scientist Friedrich Stein, and then it was called the like stromal cells or stromal stem cell. And then in 2006, 1999, it called mesenchymal progenital cell. And 2006, it became multipotent mesenchymal stromal cell. And then 2010 become mediated signal factor because it is has like a paracrine function. So here is this until 2002. This is the first time they can isolate exosome or extracellular vesicle from MSC, and then they show effect of that exosome for delivery of microRNA as anti-cancer treatment. And also here you can see a different application of MSC. So this MSC is recently called the mesenchymal stem cell. So this mesenchymal stem cell can be isolated from different sources like fat tissue. And there's two ways to culture these cells. You use explant method or enzymatic digestion. And also you can, this is very invasive method or surgical method to obtain stem cell. For example, here is a bone marrow stem cell. So you have to do invasion to the bone marrow and inside the bone and to aspiration to aspire that. So this is very painful procedure. And also, as in my lab I'm doing, we use fetal tissue for obtaining this mesenchymal stem cell. So in my lab, I used to do obtain mesenchymal stem cell from fetal tissue, especially from umbilical cord. So to understand that, if you wanted to obtain your stem cell from fetal tissue like umbilical cord, so there's many places you can obtain your stem cell from umbilical cord itself, or from amniotic membrane, and from water jelly, as I do in my lab. So this is different sources of umbilical cord stem cell. So this is my work. So this umbilical cord stem cell is very after birth, this tissue already discarded as is considered as a waste. So we can obtain that and then cut it like this and clean it by PPS and antibiotic, and clean very well and make sure to remove all blood, attach the blood from that tissue, and then we add this tissue after cleaning here in the culture plate, and then adding the kind of stem culture media and waiting until cell come out. So this method called explant method. We don't use any enzymatic digestion for obtaining this cell. So in our case, we got here also for your knowledge, these different parts to obtain stem cell from fetal tissue. So in our case, we use water and jelly. This is a very good area to get. We got here, this is our source of our mesenchymal stem cell. So this water and jelly derived mesenchymal stem cell have many advantages. It has good proliferation and differentiation compared to other source of stem cell, and also has immunosuppressive, low immunosuppressive activity because it has low expression of human leukocyte antigen class 1. And also important thing, it is fetal tissue, so you don't care about aging of the cells after you culture. For example, if you got from tissue from the elder person, you should be worried about the senescence or aging of that cells. But here, as is obtained from fetal tissue, so you should not be worried about aging or the senescence of these cells. In my laboratory, so we collaborate with hospital because our university has hospital, so we have co-work. So we co-work with the ophthalmology department, and we obtain some ocular tissue from eyes, and then we culture these cells as well, and we study. And also, I can also obtain that from mice as well. So our lab, I published this paper. So we got this paper from ocular surgery, and we got extraocular muscle and orbicularis oculi muscle. And then after culturing these cells, we obtain extracellular vesicle, and then we uh, we confirm the function of these cells as a different as a stem cell here after multilineage differentiation and the morphology, and then we tested that effect of these cells in or sorry, cell-derived EV in wound healing and so on. So if you isolate stem cell from tissue, you use some enzyme. So as I explained earlier in my slide, I use explant method, just cut and add in culture plate, and then we waiting for cells to sprout after adding culture media. Other method is for other tissue, like enzyme, like collagenase and so on. See, so here you can see different enzyme, like collagenase, and the collagenase is different type. You have to so if you use collagenase for digestion of your tissue, for obtaining or isolating MSC, you have to be careful what type of collagenase, and you have to decide your tissue and everything. That's very important to remember. And the important thing is this mesenchymal stem cell have immunomodulation function. So as you can see, this mesenchymal stem cell can have activation to some inhibition of mast cell activation, and also neutrophil and B cell, and also that's very important part for MSC function. And also MSC has two way of therapeutic action. First one is direct effect. Direct effect by differentiation. It can be differentiated to multilineage, as I mentioned before. Second way of therapeutic action is paracrine function, or it can secrete very therapeutic chemokines and growth factor and pro-angiogenic factor. This factor have itself can migrate to the injured tissue and can be have therapeutic or healing effect. That's why this paracrine function of MEC is the key idea of exosome therapeutic effect. So after obtaining your mesenchymal stem cell from tissue, whatever by explant method or by enzymatic method, whatever, you need to really make sure that cells are really MEC or not. So there is International Society for Cellular Therapy. It do some recommended some guidelines to can say this really MEC or fibroblast. So first of all, after you obtain your cells, these cells should be adherent to the plastic dish, culture dish. Second thing, there is immune surface marker or surface marker uh, must be expressed in your cells to make sure to give the term of mesenchymal stem cell or stem cell. So first thing, CD marker like CD105 should be positive, and CD73 should be positive, and CD90 should be positive. And then this marker should not be expressed, should be negative in your cells, because if you have some expression of this CD45 or 34, it means your cells are contaminated with leukocytes or blood cells. So third thing, your cells should be differentiated to multilineage, at least to adipocyte and osteoblast and chondroblast. Here the that is the name of the marker should be expressed and should not be expressed that any blood cells should not be expressed in your. That's why when you culture prepare your tissue for MSC preparation, make sure clean of the blood and washing enough to make sure not express that negative marker. So this is my work here. This cell what Angela has explained to you. So cells have spindle shape and also can have colony forming capacity. And also here markers, CD marker and stemness marker also. Negative and positive marker, and also you should make sure that marker exhibition using FACS analysis, as you can see here. So and also again, these cells can be differentiated to osteocyte and adipocyte and chondrocyte. This is the staining after we this is dyed like Alcian blue and oil red and algin red. This staining confirms that lineage differentiation. So first thing, So I give you now some overview of stem cell, types of stem cell and classes, and how to isolate stem cell and how to characterize. So this is the first thing in my I'm concerned about is promoting the therapeutic function of stem cell. So the thing I'm using is peptide mimetics. So when you culture the stem cell, we do use designed peptide growth factor derived peptide or extracellular matrix derived peptide mimetics, and then we coat the culture plate with that peptide, and then we test how this peptide can enhance the function of this stem cell. So, for example, as you can see here, there is many method or for immobilization of this peptide. Maybe you can combine this peptide with hydrogel or nanofiber or functional surfaces, or you can conjugate it with nanoparticle. And then there is many method, click chemistry or something. Some many method, chemical method. You can immobilize that peptide to whatever scaffold or culture plate in order to obtain good quality stem cell. So the concept of that, when we use, for example, FGF2 or growth factor as a recombinant protein, somehow is production is cost is very high, and then have immunogenicity. If we treat that, can have immunogenicity, and also stability is very low. So if you design some peptide that have the active part of that growth factor or protein, you can obtain high purity and also large surface area exposure and stability, and also low immunogenicity. So based on this concept, we have published paper about that. We collaborate with other company, and we designed some ECM derived peptide mimetics, for example, fibronectin and laminin motif, and we immobilize that peptide using EDCNHS chemistry, and then we find some peptide can enhance the adhesion and stemness of that induced pluripotent stem cell, as you can see here. So this among the test peptide, fibronectin derived peptide, especially PHRSN, GRDVSP, enhance the proliferation and adhesion and alkaline phosphatase activity, as you can see, of that pluripotent stem cell. This one other study, we designed some peptide about FGF. So especially FGF2, FGF has many classes: endocrine, paracrine, and so in our case, we designed the peptide related to FGF2, which is paracrine peptide, as you can see here. So important thing for FGF2, FGF2 binding need heparan sulfate to bind to receptor, but other endocrine FGF family need clotho protein to bind to receptor. So in our case, we focus on FGF2, which need heparan sulfate. So we design with other companies peptides related to FB2 or FGF2. So we screen that, make structural analysis, and we screen that in the proliferation of Watson-Jelly MSC, and we find among the test peptides, this one peptide FB2 is showing very good effect in improving the viability and also proliferation, as you can see, and also interestingly, it can increase the colony forming, and this colony forming unit or CFU is indicating the blue potency of the stem cell, and also interestingly, it can enhance the signaling pathway, like especially AKT signaling pathway, and also you could check also FRS2 alpha is upstream of FGF2 receptor cascade, and also here interestingly also this peptide can enhance the multilineage differentiation, especially the osteogenic differentiation, and also delayed the senescence here, as you can see by beta galactase staining. And not only that, we also confirm that as it enhances the osteogenic differentiation and chondrogenic differentiation, we test that in osteoarthritis model. So injecting stem cell that is cultured in an FB2 immobilized culture plate is really showing very good therapeutic effect in enhancing or healing the arthritis from collagenase type 2. So in our lab, we set up that model. We inject intra-articular administration of collagenase type 2 into the mice, and then we have to check the mouse knee joint thickness using caliper, and then we have to check the rotarod test for the behavior of mice after injection, and then test the here. Not only that, we collaborate with a very well-known scientist in Poland, and he is very famous in docking and AI technique. He also did some analysis of that receptor and peptide using his technique, and then he made very good data about that, and then he finds really good data. So so we find that our peptides really efficiently interact with receptor. So this is the conclusion of our paper. So we immobilize that peptide into the culture plate, and then we find some change in signal pathway and the mechanism of that. This is IKK and then AKT signaling pathway, and then nuclear factor kappa B. So we check at that, and then we think that can be a mechanism for enhancing the osteogenic and the chondrogenic differentiation of that. So this paper published a very good high journal over 10, and then we come here in the news in South Korea here. So second part of that work, I explained to you about tissue-derived stem cell or mesenchymal stem cell. So here I'm trying to differentiate or get this mesenchymal stem cell not from tissue, but from pluripotent stem cell. So, or this called IMSC, or induced pluripotent stem cell derived mesenchymal stem cell.